Endosperm is a tissue produced inside the seeds of most of the flowering plants following fertilization. It surrounds the embryo and provides nutrition in the form of starch, though it can also contain oils and protein. This can make endosperm a source of nutrition in the human diet. For example, wheat endosperm is ground into flour for bread. The rest of the grain is included as well in whole wheat flour, while barley endosperm is the main source of sugars for beer production. Other examples of endosperm that forms the bulk of the edible portion are coconut, meat, and coconut, water, and corn. Some plants, such as orchids, lack endosperm in their seeds. Origin of endosperm. Ancestral flowering plants have seeds with small embryos and abundant endosperm, and the evolutionary development of flowering plants tends to show a trend towards plants with mature seeds with little or no endosperm. In more derived flowering plants, the embryo occupies most of the seed and the endosperm is non-developed or consumed before the seed matures. Double fertilization. Endosperm is formed when the two sperm nuclei inside a pollen grain reach the interior of a female gametophyte, sometimes called the embryo sac. One sperm nucleus fertilizes the egg cell, forming a zygote, while the other sperm nucleus usually fuses with the binucleate central cell, forming a primary endosperm cell. Its nucleus is often called the triple fusion nucleus. That cell created in the process of double fertilization develops into the endosperm, because it is formed by a separate fertilization. The endosperm constitutes an organism separate from the growing embryo. About 70% of angiosperm species have endosperm cells that are polyploid. These are typically triploid, containing 3 sets of chromosomes, but can vary widely from diploid 2n to 15n. One species of flowering plant, Nuva polyspala, has been shown to have endosperm that is diploid, resulting from the fusion of a pollen nucleus with 1, rather than 2, maternal nuclei. The same is supposed for some other basal angiosperms. It is believed that early in the development of angiosperm lineages, there was a duplication in this mode of reproduction, producing 7-celled, 8-nucleate female gametophytes, and triploid endosperms with a 2:1 maternal to paternal genome ratio. Endosperm formation. There are 3 types of endosperm development. Nuclear endosperm formation, where repeated free nuclear divisions take place. If a cell wall is formed, it will form after free nuclear divisions, commonly referred to as liquid endosperm. Coconut water is an example of this. Cellular endosperm formation, where a cell wall formation is coincident with nuclear divisions. Coconut meat is cellular endosperm. Echoriaceae has cellular endosperm development, while other monocots are helibial. Helibial endosperm formation, where a cell wall is laid down between the first 2 nuclei, after which one half develops endosperm along the cellular pattern and the other half along the nuclear pattern. Evolutionary origins. The evolutionary origins of double fertilization and endosperm are unclear, attracting researcher attention for over a century. There are the 2 major hypotheses. The double fertilization initially used to produce 2 identical, independent embryos, twins. Later these embryos acquire different roles, one growing into the mature organism, and another merely supporting it. Thus the early endosperm was probably diploid, like the embryo. Some gymnosperms, such as Ephedra genus, may produce twin embryos by double fertilization. Either of these 2 embryos is capable of filling in the seed, but normally only one develops further. The other eventually aborts. Also, most basal angiosperms still contain the 4-cell embryo sac and produce diploid endosperms. Endosperm is the evolutionary remnant of the actual gametophyte, similar to the complex multicellular gametophytes found in gymnosperms. In this case, acquisition of the additional nucleus from the sperm cell is a later evolutionary step. This nucleus may provide the parental, not only maternal, organism with some control over endosperm development. Becoming triploid or polyploid are later evolutionary steps of this primary gametophyte. Non-flowering seed plants, conifers, cycads, ginkgo, ephedra, form a large homozygous female gametophyte to nourish the embryo within a seed. The role of endosperm in seed development. In some groups, e.g., grains of the family Poaceae, the endosperm persists to the mature seed stage as a storage tissue, in which case the seeds are called albuminous or endospermous, and in others it is absorbed during embryo development. E.g., most members of the family Fabaceae, including the common bean, Phaseolus vulgaris, in which case the seeds are called exorbuminous or cotyledonous, and the function of storage tissue is performed by enlarged cotyledons, seed leaves. In certain species, e.g., corn, zea mays, the storage function is distributed between both endosperm and the embryo. Some mature endosperm tissue stores fats, e.g., castor bean, Ricin communis, and others, including grains, such as wheat and corn, store mainly starches. The dust-like seeds of orchids have no endosperm. Orchid seedlings are mycoheterotrophic in their early development. In some other species, such as coffee, the endosperm also does not develop. Instead, the nucellus produces a nutritive tissue termed perisperm. The endosperm of some species is responsible for seed dormancy. Endosperm tissue also mediates the transfer of nutrients from the mother plant to the embryo. It acts as a location for gene imprinting, and is responsible for aborting seeds produced from genetically mismatched parents. In angiosperms, the endosperm contain hormones such as cytokinins, which regulate cellular differentiation and embryonic organ formation. Cereal grains. Cereal crops are grown for their edible fruit. Grains are caryopses, which are primarily endosperm. In the caryopsis, the thin fruit wall is fused to the seed coat. Therefore, the nutritious part of the grain is the seed and its endosperm. In some cases, e.g., wheat, rice, the endosperm is selectively retained in food processing, commonly called white flour, and the embryo, germ, and seed coat, bran, removed. The processed grain has a lower quality of nutrition. Endosperm thus has an important role within the human diet worldwide. The alurone is the outer layer of endosperm cells, present in all small grains and retained in many dicots with transient endosperm. The cereal alurone functions for both storage and digestion. During germination, it secretes the amylase enzyme that breaks down endosperm starch into sugars to nourish the growing seedling. See also: seed, ovule, beech, Chandler B. ed. 1914. Endosperm. The New Student's Reference Work. Chicago. F. E. Compton & Co. Endosperm. The pivot of the sexual conflict in flowering plants at Earthling Nature.